So based on the notions that we've seen so far, in 1991, Béliveau and colleagues published the very first example of brain imaging study with MRI. They didn't quite use oxygenated and deoxygenated hemoglobin as a contrast. So this is not a bold fMRI experiment. They used gadolinium. So the idea is that you inject gadolinium inside your participant. You wait for gadolinium to perfuse, and gadolinium flows around together with blood. So when blood rushes to a part of the brain, gadolinium will rush to that part of the brain. As I told you last time, gadolinium is paramagnetic, so it spoils the magnetic field. And so what you're seeing here is the strength of the signal on the y-axis and time. The arrow indicates when the visual stimulation appeared on screen. And what participants were doing was just staring at the blank screen, and occasionally a visual stimulation would be shown, which this arrow symbolizes. So the participant is there, the MRI signal is very strong, then the image is shown, and you see this strong dip in the signal. As gadolinium rushes to the part of the brain that is bursting with activity, the field becomes warped because of the gadolinium. And so you see the signal dips and then slowly goes back up. So this is the very first example of somebody using MRI to look at function. Of course, not with saturation of deoxygenated hemoglobin, but with gadolinium as a contrast. And what you're seeing on the left, so this image D is the easiest to look at. This is the back of somebody's head. So this is the bone and scalp, etc. So this is the back of somebody's brain. This is the right hemisphere. This is the left hemisphere. And what you're seeing here, see these colors? These colors are a statistical representation. They simply say that this part of the brain here, when the stimulation was shown compared to when it was not shown, the signal here changed significantly, changed meaningfully. Again, this is how we report fMRI results. We show which parts of the brain had the signal change together with some kind of mental activity or some kind of stimulation. So this was the very first example of MRI with gadolinium. Now, of course, not everybody is excited to the idea of getting a shot of gadolinium for an experiment. And as you know, there is an endogenous tracer inside the human brain, which is deoxygenated hemoglobin. And this was published in 1936, actually, by Linus Pauling and Charles Curiel. Now, they originally had shown that while deoxygenated hemoglobin is paramagnetic, so alters the magnetic properties of the field, the magnetic strength of the field, oxygenated hemoglobin was not. And that's because the heme groups that we discussed last lecture. So this discovery is what paved the way for doing fMRI with the bold contrast, so with the levels of oxygenation in blood as the contrast, as opposed to gadolinium. To give you an example, Fulburn and colleagues showed very nicely the relationship between how oxygenated a part of the tissue, some tissue might be, from 0 to 100%, and how quickly protons dephase. So if you look at this graph, you're going to see that the relationship says that the greater the oxygenation, towards the right of this axis, the slower protons dephase. So this is the highest oxygenation. This is 100% oxygenation. Protons take a lot of time to dephase. On the other hand, if there's little oxygenation, so on this side of the axis, then protons dephase very rapidly. So this line clearly shows this inverse relationship between the speed at which protons dephase and just how much oxygenation there is in tissue. And again, the greater the oxygenation, the slower the dephasing, i.e., the stronger the signal. Now, Seiji Ogawa was the first to realize that we could use, and this is again in the early 90s, he was the first to realize that we can actually use this fact that oxygenated and deoxygenated hemoglobin have different magnetic properties to look at brain function. And he was looking at something else. He was looking at MRI inside the animal brain, which is what you're seeing here. And he saw something peculiar in his images. He saw these little dark lines, which he was not expecting. And he realized that these dark lines are blood vessels. And see, when they're at 100%, when the brain, when the animal is breathing a 100% oxygenation solution, then these lines are very small. If now you make them breathe a 50% oxygenation solution, now the lines become very strong. And see, when Ogawa saw this of course is an artifact, right? Because if you think of it, here there's brain. You shouldn't be seeing a black line, you should be seeing a piece of brain. But he realized, and ever so cleverly, he realized that this was a contrast mechanism that could be used to study the human brain and to study its function. He immediately realized that he had on his hand a mechanism to localize changes in function, in neural function, because changes in neural functions lead to changes in metabolism and changes in level of oxygenation. So he had this brilliant intuition that these dark artifacts on his images actually would be a perfect mechanism in order to study the human brain as it functions. Just to give you an example of the same thing, but done in much higher resolution, Nico Logothetis, who at the time had a 7 Tesla animal non-human primate machine, did a very similar experiment. And in the A quadrant, you are seeing a slice of brain, and it's very oxygenated. The animal here this is a highly oxygenated slice of brain. This in B is a zoom in the lower right part of the brain, but also it has less oxygenation. And indeed, you can see these little black lines all around. See this one right here? All these black lines are nothing but blood vessels. Indeed, if you use a different imaging technique, you can see that these lines right here, for example, this one is this. So now you can really see how blood vessels are playing a key role in MRI, because these big colors that we see changing are blood vessels and the tissue surrounding the blood vessels. Now in 1992, 3 groups simultaneously published the very first human bold fMRI experiment. So the very first fMRI experiment to study function using the levels of oxygenation in brain tissue as the tracer for brain activity. And I want to show you these first 3 experiments. Now, Bandettini and colleagues, they had somebody inside an MRI machine to orient you. This image right here is a slice of the brain, and this is the back. I know it's a very poor image by today's standards. This is the back, this is the front, this is the right, and this is the left. Now what they did is they had somebody inside an MRI machine, and they would alternate periods when they would say, just rest, just relax, and that is what we call the rest or baseline. And then there would be periods when they would say, okay, now move your fingers. And that would be the active or task period. So what I want to show you on the right side is what happens to the strength of the MR signal, which is what you're seeing on the y-axis, while participants were either doing nothing, which is what you're going to see in the first block and in the last block, or when they're tapping their fingers, which is what you're seeing in the middle block. So a participant is inside the MRI machine, and we just say, just relax, do nothing. And the MR signal is around here. This is the strength of M0 in the XY plane, while the participant is just doing nothing. By the way, this is the strength of the signal in this piece of the brain right here. And then suddenly we say, okay, now move your fingers, move your fingers. And here's what you see in the bold signal. The bold signal jumps up and then stays up. What is causing this? Well, this part of the brain right here, which is marked in white because it's the part of the brain that has a significant increase in its signal during task, there must be a lot of oxygenation going to that part of the brain since the signal became stronger. Ergo, in that part of the brain, neurons must be bursting with activity, because we know that if neurons increase their activity, then there's a metabolic response. The metabolic response leads to an increase in oxygenation. If there's an increase in oxygenation, the magnetic field inside and surrounding blood vessels becomes smoother. As the magnetic field becomes smoother, protons resonate at a more similar Larmor frequency. Therefore, their signal, it takes longer for them to dephase, and their signal becomes stronger. Okay, so this is the logic of the MRI experiment. We find the piece of the brain where the signal changes between two conditions, rest and active, and we say, well, if the signal change, remember that initial picture I showed you at the beginning of this lesson, you can do it backwards. You say, well, if the signal changed between two conditions, it means that the magnetic field in this part of the brain must have changed. What leads the magnetic field to change? Well, increase in oxygenation. Okay, so what leads to increase in oxygenation? Well, a change in cerebral blood flow and cerebral blood volume. What leads to a change in cerebral blood flow and cerebral blood volume? Well, a change in metabolism. What leads to a change in metabolism? Well, neural activity. All right, so this is the chain that we draw. We just see that a part of the brain has a stronger signal, and we infer that that part of the brain must have an increase in neural activity. And so finally, we see this big increase, and then we tell the person, okay, now stop moving your hands, and the MRI signal falls off again. Right, goes back to sort of the baseline activity. So this is one of the first three experiments. Kwong and colleagues did something fairly similar, except they used visual stimulation. So instead of asking someone to move their finger, they just showed them, I believe, a simple flashing checkerboard. And they alternated periods when they were showing nothing on the screen and when they were showing a flashing checkerboard. And I know it's hard to see. What you're seeing on the left, you have to imagine that here there's a brain, and the nose is up here, and the ears are out here, and this is the back of the brain. In fact, this is visual cortex. So what this image is depicting, somewhat cryptically, is that when they're showing the flashing checkerboard, so during task, this part of the brain back here has a stronger signal. And if it has a stronger signal, we infer the underlying neural activity started increased. But when there was no flashing checkerboard shown, here the signal was not very strong. Now let's look at the exact same thing on the right. Again, this is like in the previous slide. On the y-axis, there's the signal intensity. On the x-axis, there is time. And these on and off indicate when there was nothing on screen during the off period. So this would be rest or baseline. And then the on periods, which would be task or active, when the flashing checkerboard was shown. and then off again, and then on again, et cetera, et cetera. So let's see what happens to the MR signal. So we start right here, and then you turn it on, and hoop, the MR signal goes up, then you turn off, and the MR signal falls off, and turn it on again, and hoop, it's up again. So we can clearly see how visual stimulation is clearly changing, the signal is changing in response to the visual stimulation, or at least it's changing in concomitance with the visual stimulation. So again, we infer that in that part of the brain that has its signal changing so much in the ons and offs, we infer that neurons in there are getting systematically, are increasing their activity systematically with the appearance of the visual stimulation or its disappearance. And finally, the last experiment of the three by Sergi Ogawa and colleagues, fairly similar to the one by Kwong et al., except instead of stimulating both eyes, what they did is they either stimulated the right eye or the left eye at different times with visual stimuli. And the reason for doing this is, as you might know, the right eye sends information to the left side of visual cortex, and the left eye sends information to the right side of visual cortex. So in this sense, this is a test of how precise fMRI, bold fMRI can be. So if you look on the right side, what you're seeing, this is the back of somebody's head. So this is the back, and the nose would be somewhere down here. And also, this is the left hemisphere, and this is the right hemisphere. So let's start by looking what happens in the left hemisphere. And now you can look down here. And these two lines tell you when the visual stimulation was turned on. Let's start with the bottom stimulus. Again, on the y-axis, you're seeing the strength of the T2 signal. On the x-axis, you are seeing time. So if you stimulate the right eye, remember, this is left cortex. So if you stimulate the right eye, you see that the activity, the strength of the signal is at baseline during rest. Then suddenly the visual stimulation turns on, and boom, the signal increases a lot. And the idea, if the signal is increasing, it must mean that protons are resonating more together. How do protons resonate together? Well, because the magnetic field is more smooth. What makes the magnetic field more smooth? Well, probably an increase in oxygenated hemoglobin and a decrease in deoxygenated hemoglobin. What creates an increase in oxygenated hemoglobin and a deoxygenated hemoglobin? Well, a change in metabolism. What causes the change in metabolism? Increase in neural activation, increase in neural function. All right, so that's the logic. So if you look at the left cortex, when the right eye is stimulated, when there's no stimulation, the T2 signal is at baseline. Then the stimulus turns on, which is this line. This indicates that the stimulus turns on, and you can see this big increase in activation in T2 signal. Then the stimulus turns off, and the signal goes back down to baseline, and it stays at baseline until we turn on again the stimulus, still to the right eye, and you see that the T2 signal comes up, stays up for the length of the stimulation, then the stimulus turns off again and dies down. Now, what happens to this same part of cortex if you stimulate the left eye? Turns out, not much, right? So here we're at baseline, stimulus turns on, and it just doesn't seem to react much. And then the stimulus turns off, and it doesn't change as much. And then the stimulus turns on again in the left eye, but it doesn't really affect this piece of cortex. Makes complete sense. This is the left hemisphere, the left visual cortex. So it receives mainly information from the right eye, not from the left. If you look in the right cortex, in the right hemisphere, you get the exact opposite pattern. When you stimulate the left eye, you get exactly what you'd expect. Baseline, turn on the stimulation, big response. Turn off the stimulation, it goes back down to baseline, until you turn on the stimulation again, comes back. The signal becomes strong again, turn off the stimulation, the signal dies off again. And yet again, if you stimulate the right eye, remember, this is right cortex, so it doesn't quite receive a lot of stimulation from the right eye, you see that the T2 signal doesn't change much in here, despite the presence of the stimulation. So this tells us that at least with respect to different hemispheres, the change in the bold signal is not a global change. It's not that the whole brain is changing the signal, it's changing the strength of its T2 signal. Only parts of the brain that seem to be relevant and important for whatever mental activity or task somebody is doing. So this is very important because it tells us about the spatial precision of MRI, which we'll discuss a little further next time.